Hello, good morning, students. Today we will study polysaccharides. In last lecture, we had studied carbohydrates, that is the monosaccharides, and disaccharides. Now we will study polysaccharides. What is a polysaccharide? We know polysaccharide is a carbohydrate which on hydrolysis give large number of monosaccharides, means large number of monosaccharides, they are linked by a glycosidic linkage. These are called as a polysaccharides. This examples of polysaccharides given in our syllabus are starch, cellulose, and glycogen. These are the most common natural polysaccharides. The most common natural polysaccharides are starch, cellulose, and glycogen. Starch is present in potato, then cellulose is present in all green plants, and glycogen is a polysaccharide of glucose which is present in animal body. So starch, cellulose, and glycogen, these are the common polysaccharides. Starch is a storage carbohydrate of plant, and it is an important nutrient for the humans and other animals. Starch is a storage carbohydrate of plants and is an important nutrient for humans and other animals. The second example of polysaccharide is a cellulose. It is a main constituent of cell wall of plants and the bacterial cell. It is also the main constituent of wood and cotton, and glycogen constitutes the storage carbohydrate of animals, and it is present in liver, muscles, and brain. It is also found in yeast and fungi. Now we will study structures of these polysaccharides. In polysaccharide, first polysaccharide is a starch. Starch is a polymer of alpha D glucose. Alpha D glucose, we know structure very well, that is in the glucose structure at first carbon this OH group is below the ring, that is called as alpha D glucose. This starch is composed of two components, namely amylose and amylopectin. The percentage of amylose is 15 to 20%, and percentage of amylopectin is 80 to 85%. So major constituent of the starch is amylopectin. Amylose is soluble in water and forms blue colored complex with iodine. We know this is a very famous test for the starch, and this is due to the amylose. When starch is mixed with iodine solution, then it gives a blue colored complex. So this, it contains 200 to 1000 alpha glucose units linked by alpha 1,4 glycosidic linkage, and gives unbranched chain of variable length, and this is given in figure 14.1. So first we will study the structure of amylose. This amylose consists of alpha glucose molecules. So this is the glucose molecule, we know very well. This is the carbon number 1, 2, 3, 4, 5, and 6 with this O. So this is a 6-membered ring. Here first carbon OH is below the ring, and similar structure for the glucose. So these glucose molecules are joined together by alpha 1,4 glycosidic linkage. This is first glucose molecule, this is second, this is third, this is fourth. Like this, many glucose molecules are linked together by alpha 1,4 glycosidic linkage. And this is structure of amyloids. We had studied the starch consists of 15 to 20% amyloids. It is a water soluble component and it forms the blue complex with iodine, and it contains 200 to 1000 alpha glucose molecules. They are linked together by alpha 1,4 glycosidic linkage. So here in the structure, only 4 molecules of glucose are shown. Minimum 200 and maximum 1000 molecules are linked by this type of linkage in amyloids. Now the second component of the starch is amylopectin. Exactly opposite to amyloids, it is water insoluble. Then it forms the blue violet colored complex with iodine. Then it is a branched polysaccharide chain. In amylopectin, the chains are formed by alpha 1,4 glycosidic linkage between alpha glucose units, where the branches are formed at alpha 1,6 glycosidic linkage. So two types of linkages are present in amylopectin. So amylopectin is the second component of starch. It constitutes about 80 to 85% in starch. Here, this is again glucose molecule, carbon number 1, 2, 3, 4, 5, and 6 with this oxygen. This is one glucose molecule, cyclic structure. This is other glucose molecule. So in this way, these glucose molecules are linked together by this type of alpha 1,4 glycosidic linkage. Similarly, there is a one more chain of this type of alpha 1,4 glycosidic linkage between glucose molecules, that is OH group from the first carbon and OH group from the fourth carbon. Both are below the ring. They forms a glycosidic linkage called as alpha 1,4 glycosidic linkage. So this is one chain, this is other chain. These two chains are linked together by OH group of first carbon and OH group of the sixth carbon. This is one chain, this is other chain. These chains are linked by alpha 1,6 glycosidic linkage, and this is a branch. So therefore this amylopectin is a branched component of starch. Here two types of linkages we had studied, alpha 1,4 and alpha 1,6 glycosidic linkage, and that is about the structure of amyloids and amylopectin, and hence the structure of first polysaccharide, that is a starch. Now the second polysaccharide example that we will study is a cellulose. We had already studied cellulose is present in plants. It forms the cell wall in all plants. Cellulose is a straight chain polysaccharide of beta glucose. We know what is beta glucose. In the glucose cyclic structure, if first carbon OH group is above the ring, then it is called as a beta glucose. This OH group from the first carbon of one glucose molecule and second OH group from the fourth carbon of glucose molecule forms beta 1,4 glycosidic linkage. We will study this structure. So this is a structure of cellulose. This is one glucose molecule, this is second glucose molecule, third glucose molecule. In this first glucose molecule, OH group from the first carbon, it is above the ring, and OH group from the fourth carbon, it is below the ring. So this linkage is called as a beta glycosidic linkage. This is one, this is second, this is third. Like this, many molecules of glucose, they are formed by beta 1,4 glycosidic linkage. And this type of structure obtained, that is called as a cellulose. So this implies that the beta 1,4 glycosidic bond is a very strong and difficult to hydrolyze. And this is one of the reasons why humans do not hydrolyze this cellulose. Humans do not have enzyme which can hydrolyze this linkage. Which linkage? It is a beta 1,4 glycosidic linkage that cannot be hydrolyzed by humans because enzyme is not present in the human body. Hence, cellulose cannot be digested by human being. It serves as a fibrous content of the food, useful in a bowel movement. So this Haworth projection formula for cellulose we had studied. The next example of polysaccharide is a glycogen. This glycogen has a structure similar to that of amylopectin. We had studied the structure of amylose and amylopectin. Here, the structure of amylopectin, it is a branched polymer present in starch. It is more highly branched in glycogen, and that is about the structure of polysaccharides. That is, polysaccharides given in our syllabus are starch, cellulose, and glycogen. Thank you.